Hi, my name is Sara Rouhanifard. I'm an assistant professor in the Department of Bioengineering at Northeastern University. Thank you to the organizers for the opportunity to speak today. I started my lab in January 2019, and I'm really excited to talk to you today about a direct RNA sequencing pipeline that we've been developing. So the title of my talk is Identification of Pseudouridine Modifications in mRNAs using direct long-read sequencing. So every cell in a human body comes with the same DNA, but obviously we have differential gene expression that we see in the development of different cell types and different cell states. So at the level of RNA, we have a richness in the information such as splicing, capping, and a variety of different RNA modifications that can contribute to the mechanisms that drive gene expression. So eukaryotic mRNAs are substrates for over 100 different types of RNA modification, and these are enzyme-mediated chemical modifications that can be used to fine-tune that gene expression. So the two most well-characterized examples are adenosine to inosine RNA editings, which is really important for neuronal function as well as other things, but it's very, very well known for neuronal function. And then also M6A, which interestingly has been shown to be reversible, which draws a parallel with epigenetic marks that we know a lot more about. But in general, the RNA modification field is new and there's still like a lot to learn. So pseudouridine is important for biology. It can suppress the activation of the immune system. And this is the reason why we have the 1-methyl pseudouridine that's included in the Moderna and Pfizer vaccines for SARS-CoV-2. We've seen recently that individual pseudouridine modifications are sufficient to drive splicing events. They respond dynamically to cellular stress. This includes heat shock as well as nutrient deprivation. And we also know that defective pseudourylation in cells can lead to diseases such as X-linked dyskeratosis congenita, and this has to do with the maintenance of stem cells or stemness of your cells. So pseudouridine is generated by the enzyme family called pseudouridine synthases, and in humans there are over a dozen of these that are identified so far. It's more prevalent than people realize, so it's estimated to make up between 0.2 and 0.6% of all uridines, which sounds low, but relative to everything else, it's pretty high. So it's one of the most highly abundant. M6A is the most highly abundant. Inosine is up there too, but pseudouridine is relative to the other 100 more highly abundant than the rest. We know relatively little about this particular one versus inosine and M6A that I mentioned in the previous slide for a variety of reasons. One of them, and the major one, is that there's no antibody for specific detection. And the antibody has really driven the field for M6A. So the other thing that's important is that pseudouridine does not change the base pairing properties of uridine. So you can see in this image here that pseudouridine is base pairing with adenosine. The modification, pseudouridine modification is here. There's an extra amine group added. Sorry. So it's not affecting the base pair properties. It's still binding with adenosine. And this is in contrast to inosine, that's a guanosine analog. So you're going from adenosine to inosine. So your base pairing properties have changed. So you can manipulate the Watson-Crick base pairing in order to drive hybridization. So finally, the modification is mass silent. So you can see here that the molecular weight of both of them is identical. So we can't use mass spec based methods to figure out where these are. There is a chemical called CMC, and this is a carbodiamide that labels pseudouridines twice on each of the amine groups and uridines once. It also labels guanosine once. So people have taken advantage of this for short read sequencing. So if you label your RNAs with CMC, it'll label all the pseudouridines twice, the uridines once, and after a strong base treatment, the CMC group will come off of the primary amine and it'll stay on the secondary one. So if you do cDNA synthesis, you'll get a premature stop at the site of the pseudouridine. So people have taken advantage of this for short read library prep. So what we wanted to do was to do long read sequencing so that we could detect these modifications directly, because whenever you're using a chemical, the chemical is not going to be 100% efficient. And also the strong base treatment is not really good for your RNA fidelity. And those methods are only able to pick up the very highly modified RNAs, and you can't pick up the other modifications at the same time. So we wanted to use long read sequencing So briefly, the way that this works is that your RNA is reverse transcribed to form a duplex, and it unwinds as it passes through the nanopore, and the bases, as they're passing through, are going to interrupt the current in a unique way. And that's what you're seeing here. You're seeing the current distribution as each individual base passes through the pore. And that's how you can tell the difference between different base modifications. So a uridine passing through is going to have different properties, current properties, than the pseudouridine passing through. So we were interested in seeing how quantitative we can make this method for transcriptome-wide analysis to identify sites de novo. A few other groups have shown that they can detect these base differences. So the Akison lab has shown this with ribosomal RNA, and then the Novoa group has a really awesome paper recently where they characterized the modification through nanopore sequencing as well. So our goal was really to do this transcriptome wide and identify sites de novo in a quantitative way. So when you do the long read sequencing, you can detect splicing isoforms on top of the RNA modifications. And you could also do RNA modifications like in a combinatorial way. So we could look at one modification on a single gene, or we could look at multiple modifications on a single gene. And you don't have an amplification bias that comes with this because it's direct RNA that's going through. So really briefly, the way that we do the library prep is we extract the RNA from our HeLa cells, and that's what all of the experiments that I'm going to show you are coming from. They're coming from HeLa cells. So we need another library that we know is not modified so that we can compare the modified to the unmodified and put them head to head for the exact same sequences. So using a method described by Workman et al., and the citation is down below, we reverse transcribe the RNA into cDNA, and then we apply in vitro transcription with canonical RNA bases to generate a clean RNA control with uridines in the exact same sequence context as the Psi. And it's important that it's the exact same sequence context because the nucleotides surrounding the Psi are important for how the current distribution is affected. So we chose HeLa cells because 3 other groups had previously used independent biochemical methods to identify Psi transcriptome-wide using short reads. And we wanted to validate and compare our results with these data sets. So on the top here, this is the results of our nanopore sequencing. And on the top here, so we've chosen 5 genes that have been previously validated. And we wanted to see how this looks by the nanopore. So on the top, we were seeing the current distributions for each specific 5-mer sequence that has a Psi in the center of it. So these are called K-mers. So it's a 5-mer sequence with a Psi or a U in the center of it. And we don't know how much of each we have. Like we don't know what percentage of it is Psi and what percentage is U. So in orange, we have the in vitro transcribed RNA control. So this is the clean RNA with the exact same sequence. And here we have the direct RNA sequence. And then here's a random K-mer sequence from somewhere else on the gene. So when we look at these distributions overlaid, you can see that the orange, so the clean RNA control, and the random are kind of overlapping with each other. And what you want to see, and what we do see for most of the time, is that your direct RNA reads are separate. And that's indicating that there's a difference in the current. So then when you shift over to the base calling, we see a systematic base calling error that's showing up at the site of the pseudouridine. So on the top 2 IGV plots that we have done here, you can see at the site of pseudouridine, we have a T to C mismatch or a U to C mismatch that's showing up at the site of pseudouridine. And the reason you see G on some of these, so G and A versus T and C, is because it's sense and antisense that's showing up. So in our IVT controls, we have almost no mismatch error, and we have consistent mismatch error at the site of pseudouridine for the ones that we know to be modified from previous methods. So using the mismatch percentage alone, we plotted the direct reads versus the in vitro transcribed controls. And we found that the mismatch error for the IVT, so the unmodified controls, was between 0 and 10%, which is standard nanopore error and what we expected. But we were happy to see a distribution for the direct RNA sequencing mismatch error. So what we did was we plotted all of these. So each of these dots is an individual gene that showed up by nanopore sequencing and also by in vitro transcribed RNA sequencing. So we overlaid these genes with the genes identified by 1 to 3 independent biochemical methods. And I don't have time, I won't go into each of these methods, but I've shown the general idea of what they are here. So pseudo-seq, CEU-seq, and RBS-seq. So these two are both CMC-based methods, and this one is a bisulfite RNA treatment. So we found that we could reliably detect 461 of the verified sites. The ones that we did not detect, we checked and they have very low RNA expression. So they're very low abundance RNAs that didn't get picked up by the nanopore. And we think that these were detected in the previous data sets due to the amplification that comes with the cDNA sequencing. So we were also able to identify close to 40,000 sites de novo, and we're working on validating those sites right now. So the in vitro transcribed control is a really good negative control, but generating a positive control has been really challenging for the field. So the sequence context of the SIME modification influences the current distribution. And other groups have generated some synthetic controls for this, but these have many modifications on them that also influence the current distribution. So what we need is a way to actually take a long RNA that has a single modification in the center of it and develop a model that will tell us what the current distribution looks like for pseudouridine in the sequence context that it is for a 100% modified gene. So we worked with our collaborators, Yaming Hao and Howard Gumper at Thomas Jefferson University to generate 1,000 mRNAs with a single modification in the center of them. And this is challenging to make something that's that long. I won't get into the details of how we do it. This is the scheme that we used, but we chose 4 genes that we were picking up by direct RNA sequencing. 2 of them were validated, so PSMB2 and MCM5 were validated by the other techniques. And then these 2 are 2 others that we picked up de novo. So when we ran these through the nanopore sequencer, we were also picking up the systematic base pairing error, but interestingly, but not surprisingly, when we ran it through the nanopore, not every modified site was detecting the mismatch error. So this was 100% pseudouridine that went through, and we weren't getting 100% of them with the T to C mismatch. So what we're doing now is we're taking these synthetic data sets and taking them at different ratios of 100% modified to 100% unmodified. And we're using machine learning to generate an algorithm to convert the mismatch error into a quantitative measurement of psi percentage inside of a sample for a given RNA. So the next thing we did was we took our data set and we identified 3 different types, or rather we observed 3 different types of pseudouridine modification that were showing up. And we call these type 1, type 2, and type 3. So type 1 is a high percentage of pseudouridine on a single mRNA site. And we define this as 40% and above. So if you have 40% mismatch error or above, we call this a hypermodified type 1 RNA. So an interesting observation that my student made was that when we took some of our putative pseudouridine mRNAs that were identified just by nanopore versus the one that were validated by other methods, we noticed a difference in the k-mers that were coming up. So the ones that were validated by other methods tended to be GUUCN. So the genes that were showing up, the psi was in the middle of a k-mer that was GUUCN, and ours were other k-mers. So we don't know if there's a biochemical bias that's showing up. It's something that we're looking into further. So really briefly, the other 2 types that we've noticed, they're more rare. At least type 2 is pretty rare. So type 2 is more than 1 pseudouridine possible on a single mRNA sequence. So this could mean a couple different things. It could mean that you have a single mRNA and it's modified twice at the same time on the same mRNA strand. So an example of this, you can see up here, this would be a case where you have your 2 sites. So this is the site of modification 1. This is the site of modification 2. They're separated by 3,312 bases. And when you have the mismatch showing up in both sites at the same time on the same strand, it's happening a minority of the time. The same goes for RPL15, that's only 607 bases apart. So the doubly modified ones are here. So it's a minority of the time. It seems to be that one site is the dominant modified site, but it does occur. So we call this type 2 modification. Type 3 modification is when you have a pseudouridine at one site and then another type of RNA modification on a different site. So here we have a TDC mismatch that's indicating a site of pseudouridine. And then here we have an A to G mismatch indicating an inosine. And these are on the same mRNA strand. So finally, I'm going to end with a future direction that we're working on right now. This is very preliminary data, but we were interested to see how pseudouridine modification, the percentage changes as the cell is differentiating. So we sequenced and compared psi levels before and after SHSY5-way differentiation into a neuron-like state. And we plotted neuroregulatory genes as a function of mismatch percentage between the two groups. And we noticed that there were changes in the up and down regulation for both. So this is something that we're working on for the future. This is very, very preliminary data. I just wanted to touch on it before I finish. Anyway, so just want to thank my group. Also the Wanunu lab, who is really helpful and is helping us with the machine learning for creating an algorithm to make this more quantitative. And then the HAL lab at Thomas Jefferson for helping with the synthetic controls. And the Cola-Sino lab at University of Michigan, who's helping with some lead toxicity experiments that we're following up with the SHSY5-way data. Thank you everyone for listening, and happy to answer any questions.